Hello and welcome to my presentation of the paper Learned Optical Flow for Intra-Operative Tracking of the Retinal Fundus. My name is Claudio Ravasio. I am a research assistant at KCL and a PhD student at UCL London, supervised by Lyndon Cruz from Moorfields Eye Hospital London and Christos Bergeles, who is the leader of the Robotics and Vision in Medicine group at KCL. I'll start off with some background. Vitreoretinal surgery is minimally invasive keyhole surgery inside of the eye. You can see the basic setup on the left. So little holes are made into the white of the eye in which trocars are placed, through which tools are inserted into the eye. The surgeon can see the tools through a microscope that looks through the eye's lens towards the back of the eye, towards the retina. Some of the tools used can be seen on the right-hand side: forceps, cutter, light pipe. There are even more. New procedures delivering, for example, genes or stem cells are being developed, which require higher precision than what the surgeons can currently do manually. To this end, novel robotic tools are being developed. For those to work adequately, however, we need to know the motions of the retina precisely through some computer vision-based algorithm. This is challenging, however, because of the quality of the intraoperative images. We have here two clips from our internal database called RIDE, which comprises intraoperative retinal image and video data from 66 patients, acquired during a variety of vitreoretinal surgeries at Moorfields Eye Hospital in London. So you can see glare, reflection, shadow, warping, lens effects, general movements. In the second clip, similar effects, but the lighting is even more variable and dimmer, the shadows move more quickly, the retina moves more quickly, the tool is more blurred. There's a number of effects which make tracking of any point on the retina difficult. The light reflections, as we can see here, not just on the tool itself. Double exposure can happen. Sometimes blue dye is used during a surgical procedure. It's possible to see floating fragments of membranes. Sometimes the retina is almost mirrored in the metallic shaft of the tool, which means that the tool blends into the retina a little bit. There's reflections and glare on the tool shaft. The tool themselves will cast shadows on the retina. All of this makes it difficult. So what are our general approaches? One, we use optical flow for dense motion estimation, meaning we want one motion vector per pixel. Two, we use a deep learning approach for better quality and fast inference. This is based on FlowNet by Dosovitsky et al., but with binary segmentation added in. Essentially, for each pixel, we have an output telling us whether it's part of the retina or not. And three, synthetic data for domain-specific and task-specific training. We want this algorithm to ignore occlusions such as tools, shadows, reflections, glare, and we want to predict the retinal motions underneath such occlusions, essentially impainting it from context, from visible retina and visible movements of the retina around it. So synthetic data, it must replicate the phenomena visible in intraoperative images: geometric transformations, tools, shadows, glare, even compression artifacts. To this end, we developed a flexible, extensible, complex algorithm which uses real intraoperative retinal images and tool templates as essentially raw material. It can create any required number of synthetic image pairs. Our datasets are generally the order of several 10,000 image pairs. Each image is composed layer-wise, where each layer is created individually, randomly sampling parameters for the required transformations from a uniform distribution, the limits of which are given to the algorithm as an input by the user. And for every layer, the layer-wise optical flow between the two images is calculated, which is then in the end merged down to create one final ground truth optical flow field. This is supported by a toolbox that we developed ourselves as well, which allows operations on and with flow fields, which is not always straightforward. Each record in the synthetic dataset will then in the end consist of an image pair a segmentation pair, and the ground truth optical flow. Here on the left-hand side, you can see a schematic of this layering, retina on the bottom, tool in the middle, field of view on top. In the middle we see a few sample outputs that kind of shows the varied output that we can get from the synthetic data algorithm. And on the right-hand side we see one image from some synthetic image pair and the corresponding ground truth optical flow, where you can see that the retina is moving some way and the tools are moving in a different way. Then underneath is the optical flow ground truth that we actually use for training then, where the flow of the tools is suppressed. And the idea of that is that if this is done in the training data, the algorithm will learn to essentially ignore tools and shadows, and when tested on real data, it will do the same thing. So here's an overview of our methods. We have an internal dataset that I mentioned before, RIDE. We use some material from there for our synthetic data algorithm to create the synthetic data, which again consists of image pairs, segmentation pairs, and optical flow ground truth. This synthetic data, purely synthetic data, is used to train the neural network with the usual encoder-decoder structure, which is then tested exclusively on real data. Testing benchmark sequences taken from real surgery videos recorded at Moorfields Hyde Hospital during a variety of vitreoretinal surgery procedures. So for the results, we see a table here. There's more details in the paper about how the evaluation is done exactly and what all the numbers mean. But generally, we see significant performance improvements compared to the two variational baseline methods that we compared this to. We also compared a few different network variants, and we did ablation studies for the network width, the dataset size, and the dataset complexity. It's not surprising that reducing the network width or training with less data reduces performance. It is interesting to note, however, that reducing the complexity of the dataset reduces performance even more significantly, which means that the work that we put into making the synthetic data as realistic as possible is indeed relevant for the performance achieved in the end. So the performance is maybe best seen visually here in this video. On the left, we're tracking points on a grid, and even though a tool is moving through the field of view, there's shadows, there's glare, none of that is impacting the tracking of the grid itself. And we can appreciate the flow field on the right-hand side, which is clean and doesn't show the tools in it. In comparison, the variational baseline methods here do not have a smooth flow field of that type. The retina isn't really perceived as one contiguous object. And obviously it's tracking tools and shadows individually, but it's even tracking glare patches individually. And when a tool passes in front of any point on the retina, it essentially gathers that point and moves it with it, which makes it not very useful for tracking in points of interest on the retina, whereas our method avoids this. So in conclusion, we have developed an algorithm to create large synthetic datasets of intraoperative image pairs with the corresponding ground truth optical flow field. And we have shown that optical flow learned from correctly designed domain-specific synthetic data can be used to robustly track points of interest on the retina in the presence of occlusions, shadows, tools, glare, etc. The complexity of the synthetic data has a significant influence on the achievable tracking performance. And finally, we also saw that simultaneously predicting a binary segmentation, retina, non-retina, actually helps the optical flow prediction a little bit. So this supports our initial assumption that segmentation and optical flow prediction should be two mutually supportive tasks. Thank you very much.